Hello everyone, this is Dr. Begna. I have made a video clearly explaining dental plaque composition, classification, and its formation with the help of flowcharts and hand-drawn diagrams. I have also listed out the differences between materia alba, plaque, and calculus. Everything from Carranza textbook, okay? So watch that video before this. In this video, I'll be explaining the different plaque hypotheses. So without wasting a moment, let's get into the concept. Plaque is the primary etiological factor for periodontal disease. Also, evidences support that periodontal disease act as a contributor factor for various systemic diseases like diabetes, heart diseases, lung diseases, and pregnancy-related complications like preterm low birth weight infants. To effectively treat and prevent these diseases, various hypotheses are proposed to find how healthy plaque can change into pathological plaque. Let's see the different type of plaque hypothesis. Let's see a brief description of all the plaque hypotheses. The first being non-specific plaque hypothesis. It states that entire plaque flora contributes to periodontal disease. Specific plaque hypothesis states that specific pathogens are responsible for periodontal diseases. Ecological plaque hypothesis states that microbial homeostasis imbalance results in periodontal disease. So let's see each one in detail. Non-specific plaque hypothesis. It is proposed by Miller in 1980. Its name, non-specific plaque hypothesis, was given by Loshi. This non-specific plaque hypothesis was described in the 19th century when, due to lack of technology, bacteria could not be identified. It states that periodontal disease results from the elaboration of noxious products released by the entire plaque flora. You can see this graph. Increase in plaque mass shifts the plaque flora from healthy to diseased. Can see there is a threshold level, healthy up to a certain amount, and then disease after resistance threshold. Once the body immunity is broken down by bacteria in plaque, disease occurs. When only small amounts of plaque are present, the noxious products released by the bacteria are neutralized by the host, so there is no disease. When large amounts of plaque are present, there is higher production of noxious products, which overwhelm the host defenses. The bacteria releasing toxins are defeating the host defense. So it results in disease. All plaque bacteria played a role in periodontal disease rather than any specific bacteria. Entire plaque was viewed as bad plaque. This is important in non-specific plaque hypothesis. Disease depends on amount of plaque rather than any specific pathogen. There is a plaque-induced gingivitis model in support of this theory. Lowe, Thalate, and Jensen proposed it. They examined gingival inflammation in healthy adults who ceased toothbrushing and any other oral hygiene practices. It was observed that the plaque accumulated and gingivitis developed. When plaque was removed, gingivitis started decreasing. Mechanical plaque control stopped and plaque slowly started forming. Plaque composition changed with a shift to more gram-negative species and more rods and filaments. What are the limitations of non-specific plaque hypothesis? Some individuals with considerable amounts of plaque and calculus develop gingivitis but never develop destructive periodontitis. Even when the plaque quantity is high, some developed gingivitis but it did not progress into periodontitis. The next is site specificity of plaque. In certain diseases like aggressive periodontitis, there is less calculus but there is excessive destruction. There is also site specificity in localized aggressive periodontitis where only molars and incisors are affected. Let's see the importance of non-specific hypothesis. Most therapeutic intervention was based on non-specific plaque hypothesis. This includes debridement. It may be surgical or non-surgical. This results in the removal of plaque, thereby diminishing the signs and symptoms. Let's see what specific plaque hypothesis tells us. With the invent of microscope, bacteria identification led to this hypothesis in the 20th century. This was given by Loshi. The specific plaque hypothesis underlines the importance of qualitative composition of resident microbiota. The pathogenicity of dental plaque depends on the presence of or an increase in specific microorganisms. What does this mean? Specific bacteria in plaque results in periodontal disease because that bacteria produces substances that mediate the host tissue's destruction. The key organisms produce substances which destroy the host tissues. The specific plaque hypothesis was accepted by the recognition of Aggregatibacter actinomycetum comitans as a main causative bacteria in localized aggressive periodontitis. It is also accepted by microbial complexes observed by Socransky et al. These are the color-coded complexes as suggested by Socransky. The blue complex involves various actinomyces species. Purple complex involves Veillonella, Purvula, Actinomyces odontolyticus. The green complex involves Echinella corrodens, Capnocytophagia gingivalis, Actinobacillus actinomycetum comitans. The yellow complex involves mostly Streptococcus species. The orange complex contains Campylobacter rectus, Eubacterium notatum, Fusobacterium nucleatum, Prevotella intermedia, and Prevotella nigricans. The red complex involves P. gingivalis, Tannerella forsythia, which was previously called Bacteroides forsythus, and Treponema denticola. These three complexes, the primary colonizers that get attached to tooth surfaces, are either independent bacteria, which means there are no defined complexes, or members of yellow, blue, and purple complexes. These are the primary colonizers associated with periodontal health. Secondary colonizers in plaque fell into green, orange, and red complexes. The red complex is particularly associated with bleeding or probing. The red, green, and orange complexes are associated with periodontal disease. There is certain criteria for the identification of periodontal pathogens. The first one was given by Koch and the second one was given by Socransky. Let's see the Koch postulates. The causative agent must do the following. It should be routinely isolated from deceased individuals. It should be grown in pure culture. It should produce a similar disease when inoculated into susceptible laboratory animals. It should be recovered from lesions in a deceased laboratory animal. These 4 points are important. You have to note it down. This Koch postulates can come as a very short answer question in your university exams. Let's see this. In a case of periodontitis, there are three primary problems. It is the inability to culture all the organisms because majority of organisms in periodontal disease are spirochetes. Spirochetes can't be cultured. Second one, the difficulty is inherent in defining and culturing sites of active disease. In periodontitis, there are periods of rest and periods of exacerbation. The lack of a good animal model system for the study of periodontitis. Let's see the Socransky criteria. According to these criteria, a potential pathogen must do the following. It should be associated with disease. It should be eliminated or decreased in size that demonstrate the clinical resolution of disease with treatment. That means on treatment, the bacteria should either be eliminated or decreased. The bacteria or the pathogen must induce a host response in the form of an alternation in the host cellular or humoral immune response. It should be capable of causing disease in experimental animal models. The fifth is it should produce demonstrable virulence factors that are responsible for enabling the microorganisms to cause the destruction of periodontal tissues. There are certain limitations. Studies show that periodontitis occurred even in the absence of pathogens like red or green complex in case of Socransky criteria. Sometimes these pathogens were found to be present in the absence of disease. So, there is an updated non-specific plaque hypothesis. It's a unified hypothesis. It states that whole plaque contribute to the pathogenic potential to a greater or lesser extent, but presence of some specific virulent bacteria lead to disease progression. It means all bacteria or plaque, entire plaque is responsible for producing gingivitis, but not all bacteria will lead to periodontitis. According to ecological plaque hypothesis, both the total amount of dental plaque as well as the specific microbial composition of plaque may contribute to the transition from health to disease. Periodontitis is caused by overgrowth of specific microorganisms of dental plaque when the local microenvironment changes, but it is not necessarily the same bacteria in each case. Health-associated dental plaque microbiota are stable over time and are in a state of dynamic equilibrium, leading to microbial homeostasis. Perturbations in the host response, like excessive accumulation of non-specific dental plaque, or by plaque-independent host factors like onset of an immune disorder, changes in hormonal balance as in pregnancy, or by certain environmental factors like smoking and poor diet, the number of beneficial species reduce and pathogenic species increase. This is called dysbiosis. This disturbance in ecological balance results in disease. Let's see this flowchart. It is extremely important. Ecological plaque hypothesis can come as a short answer question in your university exams. So you have to mention this flowchart. Plaque accumulation and increased inflammation lead to lower redox potential, increase in pH, increase in gingival clavicular fluid flow, higher temperature, and change in nutrients in that GCF. These ecological changes lead to transition from gram-positive microflora to gram-negative obligate anaerobes, which cause periodontitis or gingivitis. Plaque reduction and decreased inflammation will lead to higher redox potential, decreased pH, decrease in GCF flow, lower temperature. This is exact opposite of what is found in disease, right? In the absence of disease or in case of gingival health, there are predominantly gram-positive microflora, which are facultative anaerobes. Here, obligate anaerobes. These are gram-negative. You have to mention the difference. Let's see the limitation. Like other hypotheses, it does not address the role of host genetic factors that contribute to the composition of plaque and to susceptibility of periodontitis. As gingivitis don't lead to periodontitis in every case, and genetic factors can be responsible in addition to environmental factors. So, this ecological plaque hypothesis gave an additional factor like environment or ecological change, but didn't mention about the genetic changes. Let's see another important hypothesis, Keystone pathogen hypothesis. The keystone pathogen in this is Porphyromonas gingivalis. Porphyromonas gingivalis creates an environment for normal oral microbes to multiply and cause disease. P. gingivalis triggers some immune responses that lead to destruction by suppression of certain complement pathways and chemokine factors. I'll be P. gingivalis transforms normal oral microbes to dysbiotic oral microbes. This can be done by inactivating the complement system, which is leading to inflammation. So, this leads to complement dependent inflammation and bone loss. The phagocytosis is affected here. So, the keystone pathogen hypothesis was given by Hutchingalis. This hypothesis is yet to be proven in humans. This states that certain low abundance bacteria, termed as keystone pathogens, can significantly impact the oral microbiome and trigger inflammatory diseases like periodontitis, even when present in smaller quantities. Let's see the limitation. It did not explain the role of genetic factors, like the previous hypothesis. In keystone pathogen hypothesis, the main organism is Porphyromonas gingivalis. It causes three effects. The first is it causes local chemokine paralysis by blocking the production of interleukin 8 produced by gingival epithelial cells. Interleukin 8 blocking leads to delay in recruitment of neutrophils. Neutrophils can't go to the infection site properly, the infection still persists. This facilitates initial microbial colonization in the periodontium. The second effect of Porphyromonas gingivalis in keystone pathogen hypothesis is it manipulates toll-like receptor TLR response. There is decrease in level of TLR4 activity. This facilitates survival and multiplication of microorganisms in periodontium. The third effect of P. gingivalis and keystone pathogen hypothesis is it inactivates the complement system. The complement factors C3 and C5 in the presence of microbes, they cleave themselves into C3A and C5A and get activated and participate in phagocytosis of that particular microbe. P. gingivalis releases certain chemicals like gingipanes which degrade the activated forms of C3 and C5 which are responsible for phagocytosis. So this leads to loss of their function and increase in survival of microbes. By doing all these three, P. gingivalis alters the oral environment leading to disease. We have seen multiple hypotheses in this video. The non-specific plaque hypothesis states that the whole plaque is responsible for disease, while specific plaque hypothesis states that specific pathogen is responsible for causing the periodontal disease. The updated plaque hypothesis states that entire plaque is responsible for causing gingivitis, but only certain microbes are responsible for its progression into periodontitis. Ecological plaque hypothesis states that whole plaque causes gingivitis, but progression into periodontitis occurs by growth of specific bacteria by some ecological changes in biofilm. Environmental changes are taken into consideration in ecological plaque hypothesis. The keystone pathogen hypothesis states that P. gingivalis can cause periodontitis by changing the composition and increasing the quantity of microorganisms. It is a proven fact that by all hypotheses there is accumulation of plaque which is leading to gingivitis. But the progression of disease from gingivitis to periodontitis hasn't been explained clearly. Hope this is clear. This topic is very important. They can ask you a long answer question or short answer question. Each hypothesis is a separate short answer question. Pyromonas gingivalis can alone be asked as a short answer question. So be prepared, make a notes. Hope this is clear. For more such content, subscribe to my channel.